Welcome, curious minds, to the deep dive. Today, we're embarking on a really fascinating journey to truly understand the silent, intricate world of seed plants. When you gaze at something like a beautiful passion flower, its vibrant leaves reaching for the sun, its blooms attracting pollinators, you're really witnessing a masterclass in biological engineering. Even its calming properties were recognized by Native Americans. But what are the hidden, invisible forces orchestrating all that life, all that growth and development?
That's precisely our mission today. We want to pull back the curtain, so to speak, on how seed plants operate at their most fundamental levels. We're taking a deep dive into Section 6 of Physiology of Plants, 8th edition. Our goal here is to extract the essential knowledge about how these incredible organisms regulate their growth, how they exquisitely respond to their environment, efficiently acquire nutrients, and move vital substances throughout their bodies. Think of this as, well, a shortcut to understanding the hidden controls that allow a plant to thrive, focusing purely on the wisdom within our source material. No outside info today.
Okay, so we'll start by unlocking the secrets of plant hormones, those powerful chemical signals that dictate pretty much everything from a seed's decision to sprout to when a flower blooms. Then we'll explore how plants react to the world around them, from bending towards light to sensing gravity. After that, we'll dig into the ground, literally, to understand how they get their essential nutrients from the soil. And finally, we'll follow the astonishing pathways water and sugars take, often against gravity, through the entire plant. Get ready for some truly surprising facts and maybe some major aha moments about the quiet intelligence of plants.
All right, let's begin our deep dive by looking at what makes plants grow and develop in such a coordinated way. It really comes down to their internal chemical messengers, or hormones. Just like in us animals, plants use these potent chemical signals to regulate and fine-tune their metabolism, direct their growth, and shape their very form, a process we call morphogenesis. The word hormone itself, interestingly, comes from the Greek hormone, which means to stimulate.
Yeah, but what's really fascinating here is how plant hormones, often called phytohormones, they differ quite a bit from their animal counterparts, right? Animal hormones are typically produced in specialized glands and travel through the bloodstream. But plant hormones, they can be synthesized in various locations, sometimes even acting within the very same tissues where they're made or, journeying long distances. It's a more decentralized system,
Well,
but incredibly effective.
absolutely. And their potency is just incredible. We're talking about minute, almost undetectable quantities. Our source provides a fantastic analogy. The amount of a common plant hormone called indole-3 acetic acid, or IAA, found in an entire kilogram of pineapple shoot is roughly equivalent to the weight of a single needle lost in 20 metric tons of hay.
Wow, that's tiny.
It really is. And it isn't just a quirky fact. It highlights their extraordinary efficiency and the plant's exquisite sensitivity to these signals.
And that sensitivity is key, because another major difference is that plant hormones can be both stimulatory and inhibitory. So the very same hormone can trigger vastly different responses depending on the specific plant tissue, its developmental stage, or even just its concentration. It's not just about the message itself, but how the receiving cell interprets that message, which depends on its sensitivity. It's complex.
Exactly. Historically, scientists identified 5 main classes of plant hormones, often dubbed the classic 5. You've got auxins, cytokinins, ethylene, abscisic acid, and gibberellins. More recently, brassinosteroids have been recognized as a 6th major class, playing fundamental roles in development. And there are other signals, like salicylic acid or jasmonic acid, that act more specifically in defense responses. For our deep dive today, though, we'll focus on these 6 core groups, as they really govern the foundational aspects of plant life according to our text.
Okay, so let's start at the very beginning with auxin, the first plant hormone ever identified. The story behind it is quite remarkable, actually. It traces back to none other than Charles Darwin and his son Francis in 1881. They were apparently captivated by how plant shoots invariably bend towards light, that phenomenon we call phototropism.
They designed some truly elegant experiments, using canary grass and oat seedlings. Imagine taking a tiny oat seedling's protective sheath that's called the coleoptile and covering just its tip. If they covered it with something opaque, like metal foil or blackened glass, and then shone light from the side, the plant simply wouldn't bend towards the light. But if they used transparent glass instead, it would bend. This led them to a brilliant conclusion. Some influence is transmitted from the upper to the lower part, causing the latter to bend. Something was being sent down.
Right, a signal. It wasn't until 1926, though, that plant physiologist Fritz W. Wendt finally isolated this mysterious influence from oat coleoptile tips. He named it auxin, drawing from the Greek word auxin, which means to increase. The primary natural auxin, indole-3-acetic acid, or IAA, is structurally quite similar to the amino acid tryptophan.
And auxins are primarily manufactured in the rapidly growing parts of a plant. Think the nascent leaves just forming young leaves and developing seeds. They're also produced in the very tips of roots, although roots actually rely heavily on the shoot for a substantial portion of their auxin supply.
And here's one of auxin's most unique characteristics. It's the only plant hormone known to be transported polarly. That means it moves in a specific unidirectional flow, not just diffusing randomly.
Right. In shoots, this transport is always downwards, from the very tip into the stem, a process known as basipetal movement. But once it passes the root-shoot junction and heads down towards the root tip, it actually reverses direction, becoming acropetal, meaning it moves towards the apex or tip of the root.
So it goes down the stem, then toward the root tip.
Exactly. And this isn't slow, passive diffusion. It travels at a pretty impressive speed, something like 2 to 20 centimeters per hour. Our source highlights experiments where they took a segment of a seedling stem, called a hypocaule, and placed it between agar blocks. Labeled auxin quickly moves basipetally downwards into a receiver block at the bottom. But if you flip the segment upside down, very little auxin moves in what would then be the basipetal direction relative to the segment. It really proves its one-way flow within the tissue's polarity.
Okay, got it. And this polar transport is precisely controlled by special protein channels called efflux carriers, often referred to as pin proteins, named after pin-shaped mutants, right?
That's the origin, yes. Think of them as tiny, highly directional pumps. Auxin enters a cell, sometimes passively, sometimes actively, via influx carriers, and then these pin proteins, which are strategically aligned on the cell membrane, actively pump it out in the intended direction, maintaining that continuous unidirectional flow. It's like a biological conveyor belt, really.
A very specific conveyor belt.
Yeah, definitely. Auxin truly acts as a chemical maestro, coordinating growth over long distances. A classic example you probably know is apical dominance. You know how the main stem of a plant often grows vigorously upward, while the side branches or lateral buds seem inhibited? That's apical dominance.
Yeah, like in Christmas trees.
Exactly. If you snip off the shoot tip of, say, a bean plant, those lateral buds suddenly spring to life. But here's the kicker. If you apply auxin to that cut surface, lateral bud growth is inhibited once more. And it's not a direct local inhibition at the bud itself. Auxin is influencing those buds from a distance.
Wow. It also plays a pivotal role in forming the plant's plumbing system, the vascular tissues, like xylem and phloem.
Precisely. When vascular bundles are wounded, if auxin is supplied from above, it can trigger the formation of new vascular tissue, effectively reconnecting those severed lines. It's also critical for the orderly initiation and precise arrangement of leaves. Experiments with the Arabidopsis mutants, for instance, show that if auxin transport is blocked, new leaves simply don't form.
No leaves at all.
None. But a tiny micro-application of IAA, right where a leaf should be, can restore normal leaf formation. It's a testament to auxin's fundamental role in literally shaping the plant.
And for a touch of practical magic, auxin can induce parthenocarpy. That sounds fancy.
It just means producing fruit without the need for fertilization. Think seedless tomatoes or cucumbers.
Ah, okay.
Developing seeds are naturally rich in auxin. The textbook gives a vivid example. If you painstakingly remove all the tiny seeds, they're actually achens from a developing strawberry, the fruit's fleshy part, the receptacle, just stops growing. Stops dead. But apply auxin, and growth resumes normally. They even mention if you leave just a ring of achens, the fruit grows the bulging girdle just in that area, following the auxin source.
That's wild.
Isn't it? Furthermore, in woody plants, auxin kickstarts cambial activity in the spring. That stimulates the formation of new vascular tissues, essentially thickening the trunk and branches. It's also the essential signal for lateral root formation. Auxin produced at the root tip starts the process, but auxin transported down from the shoot is then needed for these new roots to actually emerge and grow out into the soil.
So auxin is really involved in almost everything related to shape and growth.
It's incredibly foundational. Now let's pivot to cytokinins. These hormones are primarily concerned with cell division and development. Our source opens this section with a compelling anecdote. Apparently, cotton growers in water-limited regions have seen yield increases of 5 to 10% just by applying cytokinins to seeds or young plants.
Really? How does that work?
Well, it stimulates root development, allowing the plant to better access water, and it even promotes a waxy leaf surface, which helps reduce precious water loss. Pretty neat adaptation.
Definitely. So what are cytokinins chemically?
They're a type of adenine derivative, structurally related to one of the bases in DNA. You find them predominantly in tissues undergoing rapid cell division. Think developing seeds, fruits, young leaves, and active root tips. They've been identified across a vast range of plant life, from simple algae to towering conifers, and even in the bleeding sap that flows from cut stems.
And are they used much commercially, like auxin?
Not quite as extensively, but they are indispensable in plant development research, especially in tissue culture and biotechnology. They are crucial for promoting shoot formation from undifferentiated cells, which is a critical step when you're trying to grow entire plants from just a few cells in a lab.
What's truly remarkable, you mentioned earlier, is their interplay with auxin.
Yes, cytokinins can often overcome that apical dominance we talked about. They can cause those lateral buds to grow even when auxin is present and trying to inhibit them.
So they argue.
You could say that. It demonstrates a sophisticated hormonal conversation within the plant, where one signal can modulate or even override another. It's all about balance.
You can apparently literally witness their anti-aging effect on leaves, too. Something about senescence.
Exactly. Senescence is just the scientific term for aging in tissues. If you take huckleberry leaves and float them on plain water, they'll typically yellow and age in about 10 days. Standard leaf aging. But add keratin, which is a synthetic cytokinin, and they stay green and fresh.
Wow.
Even more dramatically, if you apply a cytokinin solution to just a single spot on a leaf, that spot will remain vibrant green while the rest of the leaf yellows. It's like it creates a little island of youth.
And it attracts nutrients there, too.
Yeah, the text mentions it actively attracts nutrients like radioactive amino acids to the treated area. It really acts like a localized youth serum for plants.
Cytokinins also work antagonistically with auxin in the root, right? Helping maintain the root tip.
Correct. They help maintain the appropriate size of the root apical meristem, the zone of dividing cells at the root tip, and control how cells differentiate there. But in the context of lateral root initiation, cytokinin actually acts as a negative regulator. It prevents the precise auxin gradient needed for normal lateral root development, essentially putting the brakes on root branching when needed. Another example of that hormonal balance.
Okay, makes sense. Our next hormone is ethylene. You said it's a gas.
That's right, a simple hydrocarbon gas, C2H4. But it has a dramatic and surprisingly long history. Its effects were observed even before auxin was formally discovered. Imagine the 1800s, cities installing gas lights. Turns out, leaking illuminating gas was causing nearby trees to inexplicably shed their leaves, defoliate.
From the gas leak.
From the gas leak.
Sure.
It wasn't until 1901 that Dmitry Neljabov identified ethylene as the specific culprit, and it was effective even at incredibly low concentrations, as little as 0.06 parts per million.
Tiny amounts again. So what does ethylene do besides making leaves fall off?
It influences a wide array of plant growth processes. It's famous for fruit maturation, but also involved in the shedding of leaves and fruits. That's called abscission, and the overall aging or senescence of tissues. It's often a signal for change and transition in the plant's life.
There's a classic demonstration mentioned, the triple response.
Yes, in pea seedlings grown in the dark. These seedlings show 3 distinct reactions when exposed to ethylene. First, a noticeable decrease in longitudinal growth. They stop getting taller fast. Second, an increase in radial expansion. They get thicker, both in their epicotals, the stem part above the seed leaves, and their roots. And third, a horizontal orientation of the epicotals. They start growing sideways.
Why would they do that?
Picture a seedling pushing its way through compacted soil or maybe hitting a rock underground. This response helps it navigate around the debris, thicken to push harder, and change direction to find a path to the light. It's a clever survival tactic.
Makes sense. But it can also promote growth sometimes.
It can, which is interesting because it often inhibits cell expansion. But in some semi-aquatic plants, like deep water rice, it actually promotes rapid stem growth. When these plants get submerged, it triggers ethylene biosynthesis, leading to a dramatic elongation of the internodes, the stem segments between leaves. This allows the plant to keep its leaves above the rising floodwaters.
Keeping its head above water, literally.
Exactly. It also promotes the development of air spaces, called parenchyma, in the submerged tissues of other, more land-based plants, essentially helping them get oxygen down to the roots when waterlogged.
Okay, and the most famous role, fruit ripening.
Definitely. In what we call climacteric fruits, think apples, bananas, avocados, tomatoes, there's a sudden dramatic surge in respiration coinciding with ripening. This is triggered by ethylene. The fruit softens as pectin in the cell walls breaks down, and starches and acids convert into sugars, making them sweet and palatable.
Which helps animals want to eat them and spread the seeds.
Precisely. It's an evolutionary strategy. Non-climacteric fruits, on the other hand, like citrus or strawberries, ripen more gradually, without this sudden ethylene burst and respiratory peak.
And it affects sex expression in cucumbers.
Yeah, that's a surprising one. In cucurbits, that family includes cucumbers, squash, melons. High levels of another hormone, gibberellin, are typically associated with producing male flowers. But treating these plants with ethylene can shift their expression towards producing female flowers instead. It seems to do this by causing the stamens, the male reproductive parts, to undergo programmed cell death, hormones controlling gender.
Plants are just weirdly complex.
They really are. Okay, next up we have abscisic acid, or ABA. Now, its name, in a sense, is a bit misleading
Oh, so?
because it actually has very little direct role in abscission, the shedding of leaves and fruit, despite the name. It's synthesized from a carotenoid intermediate, similar to vitamin A precursors. ADA is really the plant's crucial alarm signal, especially during times of water stress, drought conditions.
Okay, so it's a stress hormone.
Primarily, yes. When conditions turn dry, the roots rapidly increase their ABA biosynthesis and release it into the xylem, the water transport system. This efficiently carries it up to the leaves.
And what happens when it gets to the leaves?
It triggers a rapid and vital response. The stomata, those tiny pores on the leaf surface that control gas exchange, respond by closing. This dramatically reduces water loss through transpiration. Our source highlights how critical this is by describing wilty mutants plants genetically unable to synthesize ABA. These mutants can only survive and grow normally in extremely humid environments where they don't lose much water. It shows ABA is essential for water conservation.
Makes sense. It's also involved in seed dormancy.
A critical player, yes. ABA acts as a natural brake, preventing seeds from germinating prematurely, maybe during a warm spell in winter, before the real spring arrives. Our source describes the viviparous VP1 maize mutants. Viviparous means they germinate while still attached to the parent plant on the cob.
Live birth, almost.
Kind of. This happens because their embryos are less sensitive to ABA, so they lack that inhibitory signal and sprout precociously. ABA ensures seeds wait for optimal environmental conditions, the right temperature, enough moisture giving them the best chance for survival. It also plays a role in inducing the synthesis of storage proteins within seeds and is vital during embryogenesis, the development of the embryo itself.
Okay, on to gibberellins, another one with a cool discovery story.
Yes. This one comes from Japanese scientists in 1926 studying a peculiar rice disease known as foolish seedling disease.
Foolish seedling.
Yeah, because the infected plants would grow incredibly rapidly, becoming tall, spindly, pale, and ultimately sickly. They look foolish, I suppose. E. Kurosawa eventually identified the cause as a substance produced by a fungus infecting the rice, a fungus called Gibberella fujikuroi, and so the substance was named gibberellin.
Caused by a fungus. So what do gibberellins normally do in plants?
They are famously responsible for the hyperelongation of shoots. They stimulate both cell division and cell elongation, particularly in stems. This is why plants treated with gibberellins can become unusually tall, or why applying it can make dwarf varieties grow to a normal size.
Is that used commercially?
It is. A major application is on table grapes. Treating them with gibberellin increases the size of the individual grapes and also loosens the clusters, making them look more appealing.
Ah. Beyond just making things taller, they also help with seed dormancy and germination.
Yes, multiple roles there.
Yeah.
In many species, gibberellins can substitute for environmental cues like cold or light requirements that are normally needed to break dormancy. They essentially enhance cell elongation, helping the embryonic root push through the restrictive seed coat. And in cereal grains like barley, gibberellins act on a specific layer called the aleurone layer, prompting it to produce enzymes like alpha amylase.
Amylase, that breaks down starch.
Exactly. It digests the stored starches in the seed into sugars, providing vital energy for the embryo's initial growth before it can start photosynthesizing.
Okay. And something called bolting.
Right. Bolting is the rapid elongation of the flower stalk. In plants that typically form low-lying rosettes, first think cabbages, carrots, lettuce, gibberellin, can induce this bolting and subsequent flowering, even without the usual trigger, which is often cold temperatures or long days. And like auxin, gibberellins can also induce parthenocarpic fruits, seedless fruits in some species, such as apples, currants, and mandarin oranges. Sometimes they're even more effective than auxin for this purpose.
Seems like there's a lot of overlap in function sometimes.
There definitely is. Hormones rarely act in isolation. It's usually a complex interplay and balance between multiple signals.
Okay, last of the big six. Brassinosteroids. These are newer.
Relatively speaking, yes. They are naturally occurring polyhydroxylated steroids. Their importance for normal plant growth became really clear when scientists studied Arabidopsis dwarf mutants that completely lack these hormones. These mutants are significantly smaller and have fewer cells than normal plants.
So essential for basic size.
Absolutely. Conversely, plants engineered to overexpress a gene called DWF4, which is involved in making brassinosteroids, grow into much larger plants than normal.
And how do they act? Locally.
It seems so. Unlike some hormones like auxin that are transported long distances, endogenous brassinosteroids appear to act more locally, right at or very near their sites of synthesis within each specific organ. Yet despite this local action, they are absolutely essential for a wide array of developmental processes. We're talking fundamental things like cell division and expansion, branching patterns, the differentiation of vascular tissues, lateral root development, seed germination, and even leaf senescence. They're involved everywhere. Our source even shows their critical role in the final maturation stage of trachery elements. Those are the vital water-conducting cells in the xylem observed in zinnia leaf cells grown in lab cultures. They needed brassinosteroids to fully mature.
Okay, so that's the main cast of hormones. But this brings us to a crucial question. How do these tiny chemical signals actually cause such profound changes in a plant? It seems like magic.
It does seem like magic, but it all boils down to regulating gene expression. Remember, plant cells are totipotent.
Meaning they can become any other type of cell.
Theoretically, yes.
Yeah.
Any single plant cell has the genetic potential to give rise to an entire new plant. This is possible because all the genes are present in every cell. But in reality, only a selected set of genes are expressed or turned on in any given cell at any given time, and that selection determines the cell's unique identity and function. Hormones are key controllers of which genes get turned on or off.
Through signal pathways.
Exactly. Hormones fundamentally alter cell growth and gene expression through what we call signal transduction pathways. Think of these as intricate biological communication networks inside the cell. They typically start with specialized protein receptors that specifically bind to a particular hormone, like a lock and key. This binding event triggers a cascade of biochemical reactions, like a chain reaction that transmits the information through the cell. Ultimately, this cascade leads to the activation or repression of specific genes by influencing key proteins called regulatory transcription factors, which control gene activity.
And these pathways can be incredibly complex, right? Almost like a tiny biological circuit board.
Absolutely, like a finely tuned domino effect. Let's consider ethylene signaling again as an example. Scientists figured this out largely by studying that triple response in Arabidopsis mutants.
Yeah.
They discovered receptors, like one called ETR1, located on a membrane system within the cell called the endoplasmic reticulum. Now, in the absence of ethylene, ETR1 actually activates another protein, CTR1, which acts as a brake, actively repressing ethylene signaling downstream.
So it keeps the ethylene response turned off normally.
Right. But when ethylene is present, it binds to that ETR1 receptor. This binding changes ETR1 shape and critically inactivates that CTR1 brake. This inactivation allows another protein, a transmembrane one called EIN2, to become active. EIN2 then transmits the signal into the nucleus, the cell's control center. Inside the nucleus, it switches on transcription factors like EIN3 and ERF1. These factors then bind to DNA and turn on or off a whole suite of genes, leading to those characteristic growth alterations of the triple response: shorter, thicker, sideways growth.
Wow, that is intricate, like a Rube Goldberg machine inside the cell.
It really is.
Yeah.
And similarly, intricate molecular machinery underpins the action of other hormones. For instance, auxin and gibberellin receptors actually function within the nucleus itself, directly influencing gene activity right there. Cytokinin receptors are transmembrane protein kinases. When they bind cytokinin, they phosphorylate themselves, add a phosphate group, and then transfer that phosphate to target proteins in a cascade, eventually activating transcription factors in the nucleus. This complex molecular dance, these specific pathways and receptors, truly explains the wide array of highly specific responses we see triggered by different hormones in different tissues.
Hormones can even influence the very direction of cell expansion. How does that work?
Yeah, that's fascinating. Imagine the plant cell wall. It's reinforced by strong cellulose microfibrils, like steel bars in concrete. If these microfibrils are deposited randomly in the cell wall, the cell tends to expand roughly equally in all directions, becoming somewhat spherical as it grows. But if these microfibrils are laid down primarily in a transverse or horizontal orientation around the cell is restricted from expanding sideways. So it's forced to expand longitudinally. It gets longer, but not much wider.
Like stretching a coiled spring perpendicular to its coils.
That's a great analogy. Gibberellins, for example, promote this transverse arrangement of microfibrils, leading to greater longitudinal growth and thus taller stems. Ethylene, on the other hand, can cause the cell's internal scaffolding, the microtubules that guide cellulose deposition, to reorient. This leads to more randomly oriented microfibrils, promoting more radial expansion, making stems shorter and thicker, as we saw in the triple response. It's all controlled at the level of the cell wall architecture.
Okay, so we've just seen how plants orchestrate their internal growth and development using this incredibly intricate network of hormones. It really is like a hidden chemical language. But what about the world outside? Plants are rooted in place, so how do they sense and react to their environment? They can't just get up and move.
Exactly. And they are far from passive. Plants are constantly reading and adjusting to a vast array of external environmental factors. We're talking about light, temperature, the length of day versus night, and even gravity. And these responses are primarily expressed through changes in their growth patterns. That's why even genetically identical plants, clones, can look very different if you grow them under varying conditions. Environment shapes form.
Right. We give a special name to these growth responses when they involve bending or curving towards or away from an external stimulus.
Tropisms. That's the term. If it bends towards the stimulus, like a shoot towards light, it's a positive tropism. If it bends away, like a root away from light, it's negative.
And the most familiar of these is undoubtedly phototropism, the way growing shoot tips curve towards light. We mentioned Darwin's early experiments with oat coleoptiles earlier.
We did, and we now know that this remarkable ability is largely orchestrated by our friend auxin.
How does light make auxin cause bending?
Well, light causes auxin to literally redistribute within the plant tip. Pioneering experiments by Winslow Briggs using coleoptiles cleverly split down the middle demonstrated this beautifully. When light shines on one side of a coleoptile tip, auxin actually migrates laterally, moving from the illuminated side over to the shaded side.
So more auxin ends up on the shady side.
Precisely. This higher concentration of auxin on the shaded side then stimulates those cells to elongate more rapidly than the cells on the sunny side. And that differential growth causes the shoot to bend elegantly towards the light source. It's not about light destroying auxin, as was once thought, but actively directing its flow.
Clever. What about gravity? That's gravitropism, right?
Correct. Gravitropism is the plant's response to gravity. If you take a seedling and lay it on its side, you'll observe its root unerringly growing downward. That's positive gravitropism, while its shoot grows upward, exhibiting negative gravitropism. They know which way is up and which way is down.
How does auxin explain that? Does it move sideways again?
It does, but the response is different in roots versus shoots. In shoots, like in phototropism, that higher concentration of auxin accumulating on the lower side stimulates rapid cell expansion there, effectively curving the stem upward against gravity.
Okay, same as towards light.
But here's where it gets really clever. In roots, which are much more sensitive to auxin, that same increased concentration on the lower side actually inhibits cell expansion.
Inhibits, so the top side grows faster.
Exactly. Meanwhile, cells on the upper side of the root with less auxin expand more rapidly. This differential growth causes the root to bend downward with gravity. It's the classic example of how different tissues can respond in opposite ways to the same hormone just because they have different sensitivities.
That's really neat. So how do they actually sense gravity? Do they have little ears or levels?
Not quote ears, but they do have sensors. It's closely linked to the sedimentation, the settling of specialized, dense, starch-containing plastids called amyloplasts. Think of them as tiny biological pebbles. These amyloplasts act as gravity sensors, or statoliths. In roots, these statoliths are found in specialized cells called statocytes, located within a region of the root cap called the columella.
So right at the very tip of the root.
Right at the tip. When a root is placed horizontally, these dense amyloplasts slide downwards within the statocyte cells, settling on the new bottom side. This physical settling somehow triggers an asymmetrical redistribution of auxin. Less auxin goes to the upper side of the root, stimulating growth there, while the higher concentration accumulating on the lower side suppresses growth, initiating that characteristic downward curve.
And specific proteins are involved in moving the auxin.
Yes, those PIN proteins again. Specifically, the text mentions PIN3 efflux carriers, which quickly reposition themselves to the lower side of the columella cells when the root is turned horizontally, directing the auxin flow downwards. Our source also points out that it's likely not just auxin. Other growth regulators, including abscisic acid, brassinosteroids, ethylene, nitric oxide, and even cytokinins, are probably implicated in fine-tuning the gravitropic response. For example, cytokinin can also become asymmetrically distributed in a horizontal root cap, potentially contributing to the growth inhibition on the lower side. It's likely a whole coordinated symphony of hormones.
Always complex. Okay, what about water? Hydrotropism.
Yes, hydrotropism is a crucial response where roots grow towards water. Obviously incredibly important for roots as they navigate the heterogeneous soil environment, seeking out patches of vital moisture.
Does auxin run that show too?
It appears to play an indispensable role, yes. But here's a curious finding. The polar transport of auxin, that strict unidirectional flow we discussed, which is so essential for gravitropism, doesn't seem to be required for hydrotropism.
Oh, so it's a different mechanism.
It suggests a distinct mechanism, or perhaps a different way auxin is involved in this specific water-seeking behavior. Roots even demonstrate positive hydrotropism when grown in microgravity, proving it's an active sensing of moisture gradients, not just passively following water downwards.
Okay. Then there's thigmotropism. That's touch, right?
Exactly, a response to physical contact. This is how roots skillfully navigate around rocks and other subterranean obstacles they bump into. Above ground, it's how climbing plants use their specialized tendrils to gracefully wrap around and secure themselves to supporting structures, like a fence or another plant.
Those tendrils can coil really fast, can't they?
Amazingly fast. Some can wrap around an object in less than an hour. The cells on the side of the tendril directly touching the support shorten slightly or grow less, while those on the opposite side elongate rapidly, creating that characteristic wrapping motion.
And they have a memory.
A short-term one, seemingly. The text mentions that pea tendrils can apparently store the memory of tactile stimulation. If you rub them, but then immediately keep them in complete darkness, they won't coil right away. However, if they are illuminated within the 2-hour window after being rubbed, they will then coil. It's like the touch stimulus is stored, waiting for a light signal to trigger the coiling response. Remarkable.
That is bizarre. Plants have memories, short ones anyway. What about internal clocks, circadian rhythms?
Yes, beyond these directional growth responses, plants also possess internal timing mechanisms. You've surely noticed how some flowers faithfully open in the morning and close at dusk, like morning glories, or how many leaves, like bean leaves, unfurl by day and refold neatly at night. This phenomenon, remarkably, was recorded way back in the 4th century BCE by observers of tamarind trees.
Wow, so these are circadian rhythms. They run on about 24 hours.
Roughly 24 hours, yes. They are regular cycles that persist even when environmental conditions, like continuous light or continuous darkness, are kept perfectly constant in a lab. They are endogenous, meaning they are driven by an internal biological circadian clock that meticulously coordinates a vast array of plant activities, things like photosynthetic activity, when stomata open and close, and even the timing of fragrance emission to attract pollinators.
And this clock is pretty robust.
Extremely. It boasts some truly amazing properties. One is temperature compensation. This allows the clock to maintain roughly the same 24-hour frequency across a range of physiological temperatures, ensuring it stays accurate whether it's a cool morning or a hot afternoon. Another property is gating. This means that an external stimulus, like a flash of light, can elicit different responses depending on when during the circadian cycle it occurs. The clock gates the plant's sensitivity to external cues.
How does the clock actually work? Genes?
Yes, it involves genes. In the model plant Arabidopsis, scientists have unraveled that this circadian oscillator involves intricate, interlocking feedback loops of specialized clock genes. Think of it like a set of interdependent genetic switches turning each other on and off. For example, light at dawn might activate certain genes, let's call them LHY and CCA1. The proteins made from these genes then act to repress another key clock gene, maybe TOC1. As TOC1's activity falls during the day, this eventually leads to a decrease in LHY and CCA1 activity. This decrease then allows TOC1 activity to rise again by dusk, which in turn might repress LHY and CCA1, restarting the cycle for the next day.
A genetic loop.
A feedback loop, exactly. And these endogenous cycles are constantly synchronized or entrained by external cues, most notably the daily cycles of light and darkness provided by the sun.
How did they figure that out? The genes involved.
Through some really clever experiments. One breakthrough involves studying the genes that encode chlorophyll ab-binding proteins, or CAB proteins, which are vital for photosynthesis and known to be clock-controlled. Scientists took the promoter region of a CAB gene, the part that controls when the gene is turned on, and fused it to a reporter gene borrowed from fireflies, the gene for luciferase, which produces light.
So the plant glowed.
Exactly. They put this construct into Arabidopsis plants. Now, whenever the plant's internal clock turned on the CAB promoter, the luciferase gene was also turned on, and the plant emitted a faint glow. They could literally see the circadian clock ticking by measuring the cycles of light emission. This powerful tool allowed them to screen for mutants with altered rhythms and identify key clock genes like TOC1.
That's brilliant. Okay, building on timing. Photoperiodism. That's about day length, right? For flowering.
Precisely. Photoperiodism is the plant's biological response to changes in the proportions of light and dark within a 24-hour cycle. This is absolutely critical because it allows plants to detect the time of year and trigger important seasonal changes, most notably initiating flowering at the appropriate time.
And there are different types of plants based on this.
That's right. Back in the 1920s, two scientists, Garner and Allard, made a crucial discovery by classifying plants into three main types based on their flowering response to day length. First, you have short day plants. These flower only when the light period is shorter than some critical length. A classic example is the cocklebur, which flowers only if the day is 16 hours long or shorter. Chrysanthemums are another common example.
So they flower in spring or fall typically?
Generally, yes. Second, there are long day plants. These flower only if the light periods are longer than their critical length. Think spinach or henbane. They usually flower in the summer.
And the third type?
Day-neutral plants. These are the easiest they flower, regardless of the day length, often based on reaching a certain size or age. Things like tomatoes and cucumbers.
Okay, but then there was a twist, right? It's not really about the day length.
Exactly. That was the really crucial insight, often called the kicker, discovered later by Hamner and Bonner. They showed it's not actually the length of the light period that's critical, but rather the length of the uninterrupted dark period that truly controls flowering in photoperiodic plants.
Uninterrupted darkness.
Yes. Take a short day plant like cocklebur again. It will flower beautifully with, say, 8 hours of light, followed by a long 16-hour continuous dark period. But if you interrupt that long dark period, even with just a brief minute-long flash of light in the middle of the night, it completely prevents flowering.
Just 1 minute of light ruins it for them.
For many short day plants, yes. Conversely, for a long day plant like henbane, which needs short nights to flower, giving it that same minute-long light flash during an otherwise long inhibitory dark period actually promotes flowering. It makes the plant think the night was short.
So the night break tricks them. And this is reversible, with different light colors.
It is. That was another key discovery. If that night-interrupting flash is red light, it has the effect we described. But if you immediately follow the red light flash with a flash of far red light, it cancels out the effect of the red light. Short day plants will flower again, and long day plants will be inhibited again.
Red versus far red. That sounds like phytochrome again.
Exactly. This photoreversibility pointed directly to phytochrome as the pigment responsible for measuring the length of the night.
And where does the plant sense this? The leaves?
Primarily in the leaves, yes. The leaf blade is where the photoperiod is perceived. You can actually induce flowering in a cocklebur plant by exposing just 1/8 of a single leaf to the correct short day cycle, while keeping the rest of the plant under long days. That single leaf tells the whole plant it's time to flower.
Okay, so phytochrome is the timer measuring the dark period.
It's the key photoreceptor. Remember, it exists in two interconvertible forms. PR, which absorbs red light around 660 nanometers and converts into the PFR form. And PFR, which absorbs far red light around 730 nanometers, can convert back to PR. PFR is generally considered the biologically active form. During the day, sunlight, rich in red light, converts most phytochrome to PFR. During the night, PFR slowly reverts back to PR or is broken down. A long night means low PFR levels by morning. A short night means high PFR levels persist. It's the level or duration of PFR that likely signals the night length.
And this PFR form influences things like germination, too.
Yes, the classic example is lettuce seeds. Many varieties require a flash of red light to germinate, which converts PR to PFR. But if you follow that red flash with far red converting PFR back to PR, they won't germinate. PFR promotes germination.
Phytochrome also explains why seedlings grown in the dark look so weird, etiolated.
Precisely. Etiolated seedlings, those grown in darkness, are long, spindly, pale yellow with unexpanded leaves. They're basically stretching, frantically searching for light. Once exposed to light, phytochrome gets converted to the active PFR form, triggering a whole suite of developmental changes called photomorphogenesis. Stem elongation slows dramatically, leaves expand, and chlorophyll is produced, turning the seedling green. It switches from search mode to growth mode.
What about plants growing in the shade of others?
Good question. Light filtered through the leaves of other plants is depleted in red light, but enriched in far red light, because chlorophyll absorbs red but transmits far red. This low red-far red ratio means less PFR is present. This signals to the shaded plant that it's being outcompeted. It triggers shade avoidance responses. The stem elongates rapidly, leaves may orient themselves more vertically, all in an attempt to reach unfiltered sunlight. If the plant senses it can't overtop its neighbors, this low PFR signal might even accelerate flowering, a last-ditch effort to reproduce before being completely shaded out.
So, if the leaves perceive the day length using phytochrome, how does that signal actually get to the growing tip, the shoot apical meristem, to tell it to switch from making leaves to making flowers? There must be a messenger.
There must be. And for decades, scientists searched for this hypothetical messenger, dubbing it phloragen.
The flower generator.
Essentially, yes.
Uh-uh.
And now, largely thanks to work in Arabidopsis, we know what it is, a protein called FT, which stands for flowering locus T, produced in specialized companion cells within the minor veins of the source leaves, is indeed the phloragen.
A protein travels all that way.
It does. It travels through the phloem, the plant's sugar transport system, journeying from the leaf up to the shoot apical meristem. Once it arrives at the meristem, it forms a complex with another transcription factor protein called FD. This FTFD complex then activates the gene that determines floral meristem identity, essentially flipping the switch and initiating the irreversible commitment to flowering. It's a remarkable long-distance protein signal that coordinates one of the most important transitions in a plant's life.
Amazing. Okay, besides day length, cold temperatures can also affect flowering. Vernalization?
Yes, vernalization is the process by which exposure to prolonged cold temperatures promotes flowering in some plants. Think of winter wheat or rye. If you plant them in the spring, they typically won't flower that summer. They just grow vegetatively.
They need the winter chill first.
Exactly. But if you plant them in the autumn, they experience the winter cold, and then they flower readily the following summer. Back in 1915, Gustav Gasner showed you could mimic this effect. If he took imbibed seeds that have soaked up water, and exposed them to near-freezing temperatures for several weeks, that rye would flower in the same summer, even if planted much later in the late spring. The cold treatment fulfilled the requirement.
So vernalization makes them able to flower, but they might still need the right day length.
Precisely. Vernalization makes the plant competent to flower, meaning it gains the ability. But they often still need a suitable photoperiod, usually long days after the cold treatment, to actually initiate flowering. This requirement for cold followed by long days is common in winter annuals, like winter wheat, and biennials, like carrots or beets that flower in their second year. It serves as a crucial protective mechanism, preventing them from flowering prematurely during a short warm spell in autumn, only to have their flowers destroyed by the subsequent winter frosts.
Where does vernalization happen? In the leaves again?
No. Unlike photoperiodism, which is perceived in the leaves, vernalization takes place directly in the cells of the shoot apical meristem itself, the growing tip that will eventually produce the flower.
And genes are involved here too.
Oh, yes. In Arabidopsis, a key gene called flowering locus C, FLC, acts as a potent inhibitor of flowering. High levels of FLC protein keep the plant in a vegetative state. Another gene, frigida, FRI, is often needed to maintain high FLC expression. Vernalization, the prolonged cold exposure, effectively works by silencing or switching off the FLC gene. This epigenetic silencing lowers FLC protein levels, removing the brake and making the meristem competent to flower when other conditions, like day length, are right.
So cold turns off the anti-flowering gene.
In essence, yes. There's even a fascinating long non-coding RNA molecule mentioned called cold air that seems to be involved in mediating the silencing of the FLC gene after prolonged cold exposure. It's like a molecular memory of winter.
And gibberellins can sometimes substitute for the cold.
Interestingly, yes. In many biennial plants, applying gibberellins can actually bypass the cold requirement, causing them to bolt, rapidly elongate the stem, and flower quickly, even without experiencing winter temperatures.
Okay, another important survival strategy. Dormancy, like hibernation for plants.
Sort of, yes. Dormancy is a special, deeply arrested state of growth. It's different from just ordinary rest, like a plant slowing down in cool weather. Dormant buds or embryos require very specific environmental cues, such as prolonged cold, a period of dryness, or maybe a specific photoperiod to be activated and resume growth. This clever strategy prevents them from sprouting or growing during superficially favorable periods, like a brief warm spell in winter, only to be killed off by a subsequent hard frost or drought. It ensures they wait for reliably good conditions.
And internal chemicals are involved. Inhibitors.
Yes. Dormant seeds or buds often contain high levels of endogenous inhibitors, internal chemicals like ABA, that actively suppress growth. These inhibitors must be either removed, for example, leached out by water, or neutralized, for example, by hormones produced during chilling, before growth can resume. Seeds have evolved various ingenious ways to break dormancy. Some desert plants have inhibitors in their seed coats that need to be washed away by sufficient rainfall, ensuring germination only happens when there's enough water. Others require mechanical abrasion of the seed coat, called scarification, maybe by tumbling in a stream or passing through an animal's gut to allow water to penetrate.
So gardeners sometimes have to scratch or soak seeds?
Exactly. Horticulturists mimic these natural processes by scarifying seeds manually or sometimes by soaking them in alcohol or even concentrated acids to break down tough seed coats.
And buds need cold too, often.
The buds of many temperate zone plant species absolutely need a period of winter cold to break dormancy. This chilling requirement is why if you cut branches from a flowering tree like a forsythia in autumn and bring them indoors where it's warm, they usually won't bloom.
Yeah.
But if you wait until late winter, after they've experienced sufficient chilling hours outdoors, and then bring them inside, they will often burst beautifully into flower. The cold broke their dormancy. Dormancy is often regulated by a finely tuned balance between growth inhibitors like ABA and growth stimulators like gibberellins.
The Doomsday Vault in Norway. That's related to dormancy.
It's a fantastic real-world application, yes. The Svalbard Global Seed Vault aims to preserve global crop diversity by storing seeds under ideal, very cold and dry conditions, maximizing their dormant lifespan. But even under these optimal conditions, they recognize that seeds slowly lose viability over decades or centuries. So the stored seeds must be periodically withdrawn, germinated to check their viability, and then grown out to produce fresh seeds for continued storage. It highlights that even deep dormancy isn't permanent.
Right. Okay, shifting gears slightly. Nastic movements. These aren't directional, like, tropisms.
Correct. Nastic movements are plant movements that occur in response to a stimulus, but their direction is independent of the direction of the stimulus. Think of a flower opening in the morning light it opens, but not specifically towards the light source.
Got it. A classic example is the sensitive plant, Mimosa pudica.
A truly spectacular example of phygmonesty, a nastic movement in response to touch. Its delicate leaflets, and even the whole compound leaf, will certainly droop and fold inward almost instantly in response to the slightest touch, a shake, or even an electrical or thermal stimulus.
How does it do that so fast?
It's due to sudden, rapid changes in turgor pressure within specialized motor cells located at the base of each leaflet and the main leaf stalk. These structures are called pulvini. When stimulated, ions like chloride, potassium, and calcium rapidly efflux, or leave, the motor cells in the pulvinus. Water follows these ions out by osmosis, causing the cells on one side of the pulvinus to lose rigidity and collapse, leading to the folding movement. It's a hydraulic mechanism.
Very cool. And related to touch, there's also thigmomorphogenesis.
Yes, that term describes how sustained, repeated mechanical stimuli, like wind, causing stems to constantly flex, or researchers rubbing stems daily in experiments, can actually alter a plant's overall growth pattern or morphology. Plants subjected to this kind of mechanical stress typically grow shorter, stockier, and more robust, with thicker stems. It's an adaptive response to make them better able to withstand the physical stress.
Like weightlifting for plants.
Kind of. Finally, many plants exhibit solar tracking, or heliotropism. This is where their leaves, or sometimes flowers, actively orient themselves to follow the sun's path across the sky throughout the day. Sunflowers are the famous example, but lupines and cotton do it too. This clever adaptation can either maximize the absorption of solar radiation, boosting photosynthesis, or in some cases, orient leaves parallel to the sun's rays to minimize heat load and water loss in hot, dry conditions. They adjust their angle based on their needs.
Okay, we've explored the intricate internal signals, the hormones, and the external responses, the tropisms and rhythms that govern how plants grow and develop. It's amazing. But none of that complex life is possible without the fundamental building blocks, nutrients. So let's shift our focus now to plant nutrition, how plants acquire these essential raw materials from their environment, how they distribute them internally, and how they put them to work for metabolism and growth.
Right, and compared to animals, plants have surprisingly simple nutritional demands, fundamentally speaking. Most green plants, the autotrophs, just need light energy, carbon dioxide from the air, and water for photosynthesis. That gives them their basic carbon structure and energy. Then, using the products of photosynthesis along with inorganic nutrients, minerals absorbed from the soil, they synthesize everything else they need, from complex amino acids and proteins to vitamins and pigments.
It's fascinating how much they can take up from the soil, though. You mentioned over 60 different chemical elements have been detected in plants, even weird stuff like gold.
It's true, they've found gold, mercury, selenium, even uranium in plant tissues. But, and this is key, not all of these are essential for the plant's survival. The presence of non-useful or sometimes even toxic elements often simply reflects the unique chemical composition of the soil they happen to be growing in. Plants absorb what's available, to some extent.
So how do we know which ones are actually essential?
The scientific pursuit to define essential nutrients really gained traction back in the 1880s, thanks to a technique called hydroponics.
Growing plants without soil? Just in water solutions?
Exactly. By growing plants in carefully formulated nutrient solutions, where scientists could precisely control which elements were present or absent, they could determine which ones were absolutely vital. Through this method, they initially identified 10 essential elements required for normal plant growth: carbon, hydrogen, oxygen, which come mainly from air and water, and then the potassium, calcium, magnesium, nitrogen, phosphorus, sulfur, and iron from the nutrient solution. Critically, if you leave out any one of these elements, the plant will fail to grow properly and will show characteristic deficiency symptoms.
And more were added later.
Yes, over time, as hydroponic techniques became more sophisticated and the chemicals used became purer, removing trace contaminants, more elements were proven essential. Manganese, zinc, copper, chlorine, boron, molybdenum were added, and then most recently in the 1980s, nickel joined the list. This brings the currently recognized total number of essential elements for most plants to 17.
17 essential ingredients for plant life. What makes an element officially essential?
There are two main criteria. First, an element is considered essential if the plant absolutely requires it to complete its entire life cycle, meaning to grow, develop, and produce viable seeds. If it can't reproduce without it, it's essential. Alternatively, an element is deemed essential if it's shown to be an integral component of some essential plant molecule or constituent. Think of magnesium, which is right at the core of the chlorophyll molecule, or nitrogen, which is fundamental to all proteins and nucleic acids. And as mentioned, the consistent appearance of specific deficiency symptoms when an element is withheld is also a very strong indicator of its essentiality.
Okay, and these 17 elements are grouped based on how much the plant needs. Macronutrients and micronutrients.
That's the standard categorization, yes.
Random.
Macronutrients are required in relatively large quantities. The threshold given is usually more than 1,000 milligrams per kilogram of the plant's dry weight. This group includes carbon, hydrogen, and oxygen, the big three from air and water, and then the mineral nutrients: nitrogen, potassium, calcium, magnesium, phosphorus, and sulfur.
So 9 macronutrients in total.
Right. Then you have the micronutrients, also sometimes called trace elements. These are needed in much smaller but still absolutely vital quantities, generally less than 100 milligrams per kilogram of dry weight. This group includes chlorine, iron, boron, manganese, zinc, copper, nickel, and molybdenum. 8 micronutrients.
Even though they need tiny amounts, they still have to actively collect them.
Often, yes. Plants actively accumulate many of these ions, frequently concentrating them inside their cells to levels much higher than in the surrounding soil solution. This movement against a concentration gradient requires energy, usually in the form of ATP.
It's interesting, too, that different plant groups might need different amounts, like eudicots versus monocots.
Yes, there are some general differences. Our source notes that eudicots, which include things like beans, sunflowers, roses, generally require higher amounts of calcium and boron compared to monocots, which include grasses, lilies, orchids, and cereals like corn and wheat. These broad patterns reflect underlying physiological differences.
Are there other elements that aren't strictly essential but are still helpful? Beneficial elements?
Yes, definitely. Beyond the core 17, some other elements are considered beneficial for specific groups of plants or under particular environmental conditions. For example, aluminum, which can be toxic to many plants, actually benefits tea plants. It seems to induce antioxidant activity and might help deter herbivores. Cobalt isn't directly essential for most plants themselves, but it is essential for the nitrogen-fixing bacteria that live symbiotically inside the root nodules of legumes. So legumes indirectly need cobalt. Sodium is essential for halophytes plants adapted to grow in high-salt environments. It also plays a role in certain specialized photosynthetic pathways, like C4 and CAM photosynthesis. And silicon provides significant structural support and enhances resistance to pests and diseases in plants like horsetails, sedges, and many grasses, It makes them tougher.
Okay, so if a plant is missing one of the essential 17, it shows deficiency symptoms. Can you tell which nutrient is missing just by looking at the plant?
Often, yes, or at least you can narrow it down considerably. Nutrient deficiencies frequently manifest as visible symptoms, especially in the plant's shoot system. Common indicators include generally stunted growth, localized patches of dead tissue, which we call necrosis, and a characteristic yellowing of the leaves, known as chlorosis, which happens because the plant can't make enough chlorophyll.
But where the symptom appears matters, right? Old leaves versus new leaves.
Absolutely critical distinction. The location of the symptoms, whether they appear first on older, lower leaves or on younger, upper leaves, provides a crucial diagnostic clue. And this location depends entirely on the element's mobility within the plant.
Mobility, meaning how easily it can be moved around inside the plant.
Exactly. Specifically, how easily it can be transported in the phloem, the plant's sugar transport system, which also redistributes some nutrients. Let's take magnesium, Mg, as an example. Magnesium is a core component of the chlorophyll molecule, and it is considered phloem mobile.
So it can be moved from old leaves to new leaves.
Precisely. If a plant becomes deficient in magnesium, it will scavenge the magnesium from its older, less productive leaves and transport it via the phloem to the younger, actively growing leaves and shoot tip, which are more vital for the plant's future. As a result, the chlorosis symptoms, the yellowing, appear first on the older leaves. Our source shows a clear picture of this in maize.
Okay, that makes sense. What about an immobile element?
Iron Phe is a classic example of a phloem immobile element. It's needed for chlorophyll synthesis, but once incorporated into older leaves, it basically gets stuck there. The plant cannot easily remobilize it and transport it to new growth via the phloem.
So if there's an iron deficiency.
The deficiency symptoms, typically chlorosis between the veins, appear first in the younger, newly developing leaves. The older leaves remain green because they still have the iron they acquired earlier. You can see this clearly in sorghum plants shown in the text.
Got it. Mobile deficiencies show up on old leaves first, immobile ones on new leaves first. What other elements fall into these categories?
Other generally phloem mobile elements include phosphorus, P, potassium K, and nitrogen N. Their deficiency symptoms also tend to appear on older leaves first. Phloem immobile ones, besides iron, include boron B and calcium Ca. Their deficiency symptoms show up on the young leaves or growing points. Understanding this mobility is really key for diagnosing plant nutrient problems.
Definitely helpful. Okay, let's turn our attention to where plants get most of these nutrients. The soil. It's more than just dirt, right?
Oh, absolutely. Soil is an incredibly complex and dynamic system. It's the primary nutrient medium for most terrestrial plants, providing not just physical support or anchorage, but also those essential inorganic nutrients, water, and even a suitable gaseous environment, oxygen, for roots to respire. Understanding soil properties is absolutely fundamental, especially for agriculture and ecology.
And soils have layers, horizons.
Yes. Mature soils typically develop distinct horizontal layers called horizons. The uppermost layer, the A horizon, is commonly known as topsoil. This is the zone of most intense physical, chemical, and biological activity. It's usually rich in decomposed organic material, which forms humus, giving it a dark color, and it absolutely teems with life. Plant roots, insects, earthworms, protists, nematodes, fungi, bacteria, a whole ecosystem. Earthworms, for instance, are amazing soil engineers, processing vast quantities of soil, aerating it, and depositing highly fertile castings.
Humus and clay are important for holding water and nutrients.
Very important. The soil's ability to hold water is crucial. We talk about field capacity, which is the maximum percentage of water a soil can hold against the pull of gravity after excess water is drained away. Clay soils, because they have much finer particles and smaller pore spaces, can hold significantly more water than sandy soils, which have large particles and drain quickly. But plants can't get all the water held in soil. When the soil water potential drops so low, the book says to about -1.5 megapascals, that the plant cannot exert enough force to extract water, the permanent wilting percentage is reached. At this point, the plant will wilt and likely won't recover unless water is added.
And soils hold on to nutrients too, especially positive ones.
Yes, particularly cations, those positively charged ions like calcium Ca2+, potassium K+, magnesium Mg2+, and ammonium NH4+. These cations are attracted to and held by the negatively charged surfaces of tiny clay particles and humus molecules in the soil. This is called anication exchange capacity. This binding prevents these essential cations from being easily leached or washed away by water percolating through the soil. However, anions negatively charged ions like nitrate NO3, sulfate SO42, and chloride COY, are generally repelled by the negative soil charges and are therefore more susceptible to being leached out, potentially polluting groundwater.
What about iron? You said it's often hard for plants to get.
Right, iron is actually very abundant in the Earth's crust, but in well-aerated soils, it's typically found in the oxidized ferric Fe3+ form, which is highly insoluble, especially at neutral or alkaline pH. So even though there's lots of iron around, it's often unavailable for plants to absorb.
So they have special tricks to get it. Strategies.
They do. Plants have evolved two main strategies. Most plants, including eudicots and non-grass monocots, use what's called strategy 1. They actively pump protons, H+, out of their roots, acidifying the soil immediately around them, the rhizosphere. This acidity helps dissolve F3+ form. They also have enzymes on their root surface that reduce the F3+ to the more soluble ferrous, Fe2+ form, which can then be transported across the root cell membrane by specific transporter proteins.
And strategy 2 is for grasses.
Primarily, yes. Grasses use strategy 2. Instead of just acidifying, they release specialized organic molecules called phytocytophores into the soil. These molecules act like chelators, think of tiny chemical claws that bind very tightly to the insoluble F3+ willage. The entire FF3+ to phytocytophore complex is then absorbed by the grass root through specific transporters. Two different clever solutions to the same problem.
And soil pH matters a lot for nutrient availability in general.
Hugely important. As we just saw with iron, solubility changes with pH. In alkaline, high pH soils, many essential locations, including iron, manganese, copper, and zinc, can precipitate out as insoluble hydroxides or carbonates, making them unavailable to plants even if they're physically present in the soil. Conversely, in very acidic, low pH soils, elements like aluminum and manganese can become too soluble, reaching toxic levels. So there's an optimal pH range for most plants, usually slightly acidic to neutral.
And you mentioned mycorrhizas before the fungi.
Yes, mycorrhizas are these incredibly important symbiotic associations between fungi and plant roots found in the vast majority of plant species. The fungal hyphae extend far out into the soil, much farther than the plant's roots can reach, acting like an extension of the root system. They are particularly effective at absorbing nutrients that are relatively immobile in the soil, especially phosphorus, but also zinc, copper, and manganese, and transferring them back to the plant. In return, the plant provides the fungus with sugars from photosynthesis. It's a crucial partnership for nutrient acquisition in many ecosystems.
Okay, so plants get nutrients from soil, but those nutrients have to come from somewhere and get recycled. Nutrient cycles.
Exactly. Since Earth is fundamentally a closed system regarding matter, with a finite supply of each element, life as we know it absolutely depends on the constant recycling of these essential nutrients. These grand recycling pathways are called biogeochemical cycles because they involve a continuous interplay between biological organisms, biogeological processes, and chemical transformations, chemical.
And some cycles are global, others more local.
That's a key distinction. Cycles involving elements that have a significant gaseous phase in the atmosphere, like carbon CO2, oxygen O2, sulfur SO2, H2S, and especially nitrogen N2, are considered global cycles because the atmosphere allows for rapid worldwide distribution. Other cycles, for elements that don't really have a gaseous form, like phosphorus, calcium, and potassium, tend to be more localized. They primarily cycle within the soil, water, and living organisms within a particular ecosystem or watershed.
But these cycles aren't perfect. They can be leaky.
They definitely can be. Natural processes like soil erosion can wash nutrients away, and human activities have greatly exacerbated these losses. Things like harvesting crops, which removes nutrients embodied in the plants, burning forests or grasslands, which releases nutrients to the atmosphere or in ash that can be washed away, and agricultural practices that lead to increased leaching all contribute to the loss of precious nutrients from local ecosystems, potentially depleting soil fertility over time.
Let's talk about nitrogen specifically. You said it's often the most limiting nutrient, even though the air is full of it.
It seems paradoxical, doesn't it? The atmosphere is the vast reservoir, about 78% nitrogen gas, N2. But the problem is that N2 molecule has an incredibly strong triple bond holding the two nitrogen atoms together. Most living organisms, including all plants and animals, simply cannot break that bond to use the N2 directly. They depend entirely on more chemically reactive forms of nitrogen, primarily ammonium, NH4+, and nitrate, NO3, which are present in the soil but usually in much, much lower concentrations than the N2 in the air. That scarcity of usable nitrogen is why it so often limits plant growth in both natural ecosystems and agriculture.
So the nitrogen cycle is all about converting that unusable N2 into usable forms and recycling it.
That's the core of it, yes. It's the complex process by which this limited but absolutely vital usable nitrogen is circulated and transformed through the environment and living organisms. It involves several key stages and types of microorganisms.
What's the first step in recycling nitrogen from dead stuff?
That would be a modification, also sometimes called nitrogen mineralization. When plants and animals die, decomposers, primarily saprotrophic bacteria and fungi in the soil, break down the complex organic nitrogen compounds, like proteins and nucleic acids, in the dead material. In doing so, they release the nitrogen back into the soil in the form of ammonium ions, NH4+.
Okay, so decomposition makes ammonium. What happens next?
Next comes nitrification. This is usually a two-step process carried out by specialized groups of soil bacteria. First, bacteria like Nitrous amonis oxidize the ammonium, NH4+, into nitrite ions, NO2. This nitrate is actually quite toxic to most plants. Fortunately, a second group of bacteria like Nitrobacter rapidly oxidizes the nitrite, NO2, further into nitrate ions, NO3. Nitrate is the primary form of nitrogen that is readily absorbed and utilized by the roots of most crop plants and many plants in natural ecosystems. So nitrification is crucial for making nitrogen available in the preferred form.
And then some gets lost back to the air. Denitrification.
Correct. Denitrification completes the cycle by returning nitrogen to the atmosphere. Under anaerobic conditions, situations where oxygen is scarce, like in waterlogged soils or sediments, certain bacteria can use nitrate as an alternative electron acceptor for respiration, instead of oxygen. In this process, they convert nitrates, NO3, back into gaseous forms of nitrogen, primarily nitrogen gas, N2, but also sometimes nitrous oxide, N2O, which is a potent greenhouse gas. This gaseous nitrogen then escapes back into the atmosphere.
So nitrogen is constantly cycling, fixed from the air, incorporated into organisms, decomposed to ammonium, nitrified to nitrate, absorbed by plants, and some denitrified back to the air, plus losses through erosion and harvesting.
You've got it. It's a dynamic balance. And because usable nitrogen is often scarce and easily lost, the process that replenishes it is fundamentally important.
Which is nitrogen fixation, getting it from the air.
Exactly. Nitrogen fixation is the crucial process where inert atmospheric nitrogen gas, N2, is reduced, chemically converted into ammonium, NH4+, a form that can finally be incorporated into organic compounds by living organisms. This incredibly energy-intensive reaction is catalyzed by a remarkable enzyme complex called nitrogenase. And here's the amazing part. Nitrogenase is found only in certain types of bacteria and archaea. No eukaryotes, no plants, no animals, no fungi can fix nitrogen on their own. We are all utterly dependent on these microbes.
And some of these microbes team up with plants, legumes.
Yes. The most well-known and often most effective nitrogen-fixing bacteria are those that form intimate symbiotic relationships with plants, particularly with members of the legume family that includes peas, beans, soybeans, clover, alfalfa, peanuts, etc. In this incredible mutualistic partnership, the bacteria, commonly called rhizobia, live inside specialized structures called root nodules on the plant's roots. The bacteria fix atmospheric N2 into ammonia for the plant, providing it with a direct source of nitrogen. In return, the plant provides the bacteria with carbohydrates, sugars, from photosynthesis for energy, and a protected low-oxygen environment because nitrogenase is actually inactivated by oxygen.
People knew about this benefit for a long time, like the Greeks.
Absolutely. Ancient Greeks and Romans and farmers throughout history recognized the value of growing legumes in crop rotations, or as green manure, plowing the legume plants back into the soil to enrich the soil and improve the yield of subsequent crops. They knew it worked, even if they didn't understand the microbial basis until much later.
How do these nodules actually form? It sounds complicated.
It is a very sophisticated, genetically controlled process involving signaling between the plant and the bacteria. It typically starts with the plant roots releasing specific chemical signals like flavonoids into the soil. These attract compatible rhizobia bacteria to the root hairs. The bacteria, in turn, produce signaling molecules, nod factors, that cause the root hair to curl around them. The bacteria then induce the formation of an infection thread, essentially an invagination of the plant cell wall and membrane, which grows inwards, carrying the dividing bacteria into the root's outer layer, the cortex. Simultaneously, these bacterial signals stimulate cortical cells nearby to start dividing rapidly, forming a nodule primordium, the beginning of the nodule structure. Eventually, the bacteria are released from the infection thread into the cytoplasm of the plant cells, but they remain enclosed within a plant-derived membrane, forming structures called symbiosomes. Inside these symbiosomes, The bacteria differentiate into specialized, often enlarged forms called bacteroids, which are the actual nitrogen-fixing factories.
Wow. A lot of coordination. Are all nodules the same?
No, there are two main structural types. Indeterminate nodules, like those found in alfalfa, peas, and clover, are typically elongated or cylindrical and possess a persistent meristem at their tip. This means they continue to grow throughout their lifespan. Determinate nodules, like those in soybeans, common beans, and lotus, are typically spherical and lack a persistent meristem. Their growth stops relatively early.
And hormone balance is critical for this.
Extremely. Nodule formation is tightly regulated by a complex interplay of plant hormones. Auxins, cytokinins, gibberellins, and brassinosteroids generally act as positive regulators, promoting nodule development. Conversely, hormones often associated with stress or defense responses, like abscisic acid, ethylene, jasmonic acid, and salicylic acid, tend to act as negative regulators. This intricate hormonal control likely prevents the plant from forming too many nodules, which would be a huge drain on its energy resources. Remember, nitrogen fixation is very costly. It's a carefully managed symbiosis.
Do only legumes form these nitrogen-fixing nodules?
Mostly, but not exclusively. There's a tropical tree genus called Parasponia, which remarkably forms nodules with rhizobia, even though it's not a legume. And other plants, like alder trees, Alnus and Bayberry america, form nitrogen-fixing nodules with different types of bacteria called Frankia, which are actin-mycetes. Even more broadly, some nitrogen-fixing bacteria, including certain rhizobium strains and others like Azorchis, can live inside the tissues of non-nodulating crops, such as barley, maize, rice, and wheat. They live as endophytes, residing in intercellular spaces or even within the xylem vessels. These associations can provide substantial amounts of nitrogen to the host crop that text mentions, potentially up to 60% or even more.
That's huge. And then there are the weird ones, carnivorous plants.
Right. For a truly wild biological adaptation, some carnivorous plants like bladderworts that trap aquatic organisms or butterworts with sticky leaves that trap insects have essentially bypassed a soil nitrogen cycle in nutrient-poor habitats like bogs. They obtain their nitrogen, and sometimes phosphorus, directly by digesting the animal protein from the prey they capture. A very different strategy.
Okay, so once the plant gets nitrogen, either fixed or absorbed as nitrotimonium, how does it use it? Assimilation.
Yes, assimilation is the process of incorporating that inorganic nitrogen into organic molecules. Once absorbed, nitrate is usually reduced back to ammonium, and then this ammonium is rapidly incorporated into amino acids, primarily via a key metabolic pathway called the glutamine synthetase glutamate synthase, GS-GROGAT pathway. This often happens in close association with photosynthesis in the leaves, or it can occur in root plastids using energy derived from respiration.
And humans now fix nitrogen too, industrially.
Massively. The Haber-Bosch process, developed in the early 20th century, is an industrial method for synthesizing ammonia, NH3, directly from nitrogen gas, N2, and hydrogen gas, H2, typically derived from natural gas, methane. This process now produces something on the order of 50 million metric tons of fixed nitrogen fertilizer annually. That's enormous. It's nearly half as much as all the biological nitrogen fixation occurring naturally across the entire globe each year.
Yeah.
It's been credited with sustaining food production for a large part of the world's population.
But it's energy intensive.
Very. The Haber-Bosch process requires extremely high temperatures, around 400–500 degrees C, and pressures, 150–350 atmospheres, plus catalysts, to break that strong N2 triple bond. This consumes vast amounts of energy, mostly derives from fossil fuels, making it a significant contributor to greenhouse gas emissions. Biological nitrogen fixation, while complex, happens at ambient temperature and pressure.
Okay, let's touch on phosphorus, also crucial, but needed in smaller amounts than nitrogen.
Generally, yes, plants need less phosphorus than nitrogen, but phosphorus is very often the most limiting nutrient that plants derive from the Earth's crust, especially in older, highly weathered soils like those found in Australia or the tropics. Why? Because phosphorus is typically present in very low concentrations in the soil solution, it's distributed very unevenly, and it's almost immobile in soils. It doesn't move readily to the plant roots. The roots have to grow to find it. This combination makes it hard for plants to acquire enough.
And its cycle is more local, no gas phase.
Correct. The phosphorus cycle is much more localized than the nitrogen cycle because phosphorus doesn't have a significant gaseous form to enter the atmosphere. The primary reservoir is phosphate minerals in the Earth's crust. Phosphorus cycles from rocks to soil through slow weathering, gets taken up by plants, moves into animals that eat plants, is returned to the soil as organic forms in dead organisms and waste products, and then soil microorganisms mineralize that organic phosphorus back into inorganic phosphate, PO43, the main form plants can absorb. Losses occur primarily through erosion and leaching and are replenished very slowly by the weathering of rocks.
So plants need special ways to get phosphorus too, since it's so scarce and immobile.
They absolutely do. They've evolved several clever strategies to efficiently scavenge for phosphate. One is simply architecture, developing extensive root systems with many fine lateral roots and abundant root hairs, dramatically increasing the surface area for absorption. Another, as we mentioned, is forming those symbiotic relationships with mycorrhizal fungi. The fungal hyphae act like a vast extension of the root system, exploring a much larger volume of soil and effectively mining for phosphorus than delivering it to the plant.
And some plants have those special roots, cluster roots.
Yes, some plants, particularly those adapted to extremely phosphorus-poor soils like many species in the Proteaceae family from Australia, Echihachia, develop remarkable structures called cluster roots or proteoid roots. These are dense clusters of short, densely packed lateral rootlets covered in root hairs, looking almost like a bottle brush. They massively increase the root system's absorbing surface area in localized soil patches. But they do more than just increase surface area. These cluster roots also release large quantities of organic acids, like citrate and malate, along with phosphatases, enzymes that release phosphate from organic matter, and protons, acidifying the soil, in a coordinated exudative burst. This chemical cocktail effectively mobilizes phosphorus that's locked up with soil minerals like iron, aluminum, or calcium, making it soluble and available for uptake. It's a very active foraging strategy.
Plants are really resourceful, but human activities are messing up these cycles, right?
Unfortunately, yes, quite significantly. Our activities, largely driven by the immense pressure to feed a growing global population, have drastically altered natural nutrient cycles. Intensive agriculture, involving crop removal year after year, takes large amounts of nutrients like phosphorus and nitrogen out of the system. Increased soil erosion due to deforestation and poor land management washes nutrients away. We're also destroying natural ecosystems like marshes and wetlands, which are crucial natural sites for processes like denitrification, further disrupting the nitrogen cycle balance.
And fertilizer runoff is a big problem. Nitrate pollution.
A huge problem in many agricultural regions. Excess nitrogen fertilizer applied to fields, especially nitrate, which is very mobile, often isn't fully utilized by crops. It leaches into groundwater and runs off into rivers and lakes. This nitrate pollution contaminates drinking water sources. The text specifically mentions widespread contamination in California, which can pose health risks, particularly to infants. It also causes eutrophication of aquatic ecosystems, algal blooms that deplete oxygen and harm fish life. The source even mentions a potential future threat. Vast reservoirs of nitrate that have accumulated naturally in deep desert subsoils over millennia could become mobilized and contaminate groundwater if these arid lands are irrigated for agriculture or if climate patterns shift significantly.
So research in plant nutrition is really important for finding solutions.
Incredibly valuable, yes. Scientists are actively working on multiple fronts. One approach is using traditional plant breeding and modern biotechnology to develop new crop cultivars that are inherently more efficient at acquiring nutrients from deficient soils, or that are more tolerant to toxic levels of certain elements found in problematic soils, like acidic soils or mine tailings.
Like plants that can clean up pollution. Phytoremediation?
Exactly. Phytoremediation is a fascinating field that uses plants to remove pollutants from contaminated soil or water. One strategy involves using hyperaccumulators. These are unusual plants that have evolved the ability to absorb and concentrate extremely high levels of heavy metals like zinc, nickel, cadmium, lead, even arsenic in their shoots, often thousands of times higher than normal plants, without suffering toxic effects
They
themselves.
store the metals.
They transport them to their shoots and store them, often safely sequestered in cell vacuoles or bound to specific molecules, frequently in leaf epidermal cells or hair-like structures called trichomes, which minimizes damage to their vital photosynthetic cells in the leaf interior. These hyperaccumulators, many found in the mustard family, Brassicaceae, can literally be grown on contaminated sites, harvested, and then the metal can be recovered or safely disposed of. They can potentially clean up decades of industrial metal pollution from soils. It turns out they achieve this feat largely by overexpressing genes that are common to all plants, but involved in metal uptake, transport, and sequestration. They just turn the volume way up on these normal processes.
What about aluminum toxicity? You mentioned that's common in acid soils.
Yes, aluminum toxicity affects a huge proportion of the world's arable land, maybe 30–40%, particularly in the tropics and subtropics, where soils are often naturally acidic and highly weathered. In acidic soils, aluminum becomes soluble as the toxic Al3+ ion, which severely inhibits root growth, limiting water and nutrient uptake. Some plants have evolved resistance. Their primary mechanism is to efflux or pump out organic acids, particularly citrate and malate, from their root tips. These organic acids bind very strongly to the toxic Al3+ ALA, forming non-toxic complexes in the soil right outside the root, effectively preventing the aluminum from being absorbed into the sensitive root apex.
Clever defense. And finally, improving nitrogen fixation.
That holds immense potential for more sustainable agriculture, reducing our reliance on energy-intensive Haber-Bosch fertilizers. Researchers are working on several angles. They're screening existing legume varieties and rhizobial strains to find the most efficient symbiotic partnerships. They're developing biofertilizers, inoculants, containing highly effective nitrogen-fixing bacteria to improve associations with both legumes and potentially those non-nodulating crops that harbor endophytic fixers. But the most ambitious long-term goal, often considered the holy grail of this field, is genetic engineering. The ultimate aim is to transfer the entire set of genes required for nitrogen fixation, the NIF genes from nitrogen-fixing bacteria, into non-fixing bacteria that associate closely with crops, or even eventually directly into the genomes of major crop plants like corn, wheat, or rice, enabling them to fix their own nitrogen from the air. That would be truly revolutionary.
All right, we've journeyed through hormones, environmental responses, and nutrient acquisition. It's been fascinating. But a crucial piece of the puzzle remains. How do plants actually move all these vital ingredients, the water and minerals from the soil, and the sugars they produce in their leaves, to every single cell where they're needed, often over long distances? This is the story of internal transport.
Exactly. Plants are absolute masters of internal plumbing. They efficiently transport both organic nutrients, primarily sugars, and inorganic nutrients, minerals, alongside vast quantities of water throughout their entire bodies. They achieve this using a highly specialized and continuous vascular system made up of two main tissues, the xylem and the phloem.
Xylem for water, phloem for food, right?
Yeah.
That's the basic idea.
That's the primary distinction, yes. Xylem is mainly responsible for the bulk transport of water and dissolved minerals from the roots up to the rest of the plant. Phloem primarily transports sugars produced during photosynthesis, the assimilates, from the leaves, sources, to other parts of the plant where they're needed. Sinks. But it's important to remember their functions often overlap significantly. For instance, phloem actually moves considerable volumes of water along with the sugars. It even provides the main water supply to developing fruits, which often have poor xylem connections. And there's constant exchange of water and solutes between the xylem and phloem systems. They work together.
People were curious about this internal movement for a long time, even back in the 17th century.
Oh, absolutely. Early investigators were fascinated by this apparent circulation within plants. Stephen Hales, a famous English clergyman and scientist in the 18th century, conducted some ingenious experiments. He famously calculated that a single, modest-sized sunflower plant could imbibe and perspire, essentially take up water through its roots and lose it through its leaves, an astonishing 17 times more water by weight than a human does in the same 24-hour period.
17 times more. That's an enormous amount of water passing through the plant.
It really is. And it highlights a critical point made in the text. Nearly 99% of all the water a plant absorbs through its roots is ultimately lost to the atmosphere as water vapor. Only a tiny fraction is actually used for growth or photosynthesis.
99% lost. Why does that happen? What's that process called again? Transpiration.
Exactly, transpiration. It's this essentially unavoidable loss of water vapor from the plant, mostly through the leaves. And while it's a necessary consequence of being a photosynthesizing land plant, it can also be extremely detrimental if water is scarce, leading to dehydration, wilting, and significantly reduced growth.
So why is it unavoidable? Why do they lose so much water?
It boils down to a fundamental conflict, a design trade-off inherent in land plants. To capture enough sunlight for photosynthesis, leaves need to have a large surface area. That's obvious. And for photosynthesis to happen, the plant needs to take in carbon dioxide, CO2 from the atmosphere. For CO2 gas to enter the plant cells inside the leaf, it must first dissolve into the moist surface of those cells.
So the inside of the leaf has to be wet.
Essentially, yes.
Okay.
There needs to be a wet surface for gas exchange. But here's the problem. Wherever water is exposed to air that isn't fully saturated with water vapor, which is most of the time, evaporation will occur. Water molecules will escape from that moist surface into the drier air. So the plant's essential need to open up its leaves to let CO2 in for photosynthesis inevitably means that water vapor will escape out. Carbon dioxide uptake and water loss through transpiration are thus inextricably linked.
a necessary evil then. But plants can control it somewhat. They're not just leaky sieves.
No, they're definitely not passive victims. They've evolved sophisticated ways to control water loss. First, the entire outer surface of their leaves and young stems is covered by a waxy layer called the cuticle. This cuticle is largely impervious to both water and CO2. Only a very small fraction of the total water loss, maybe 5–10%, occurs directly through the cuticle, cuticular transpiration, or through small pores called lenticels found in the bark of older stems.
So where does most of the water escape from? The stomata.
Exactly. By far the largest amount of transpired water, often over 90%, is lost through thousands of tiny adjustable pores on the leaf surface called stomata. Each stoma is essentially a pore flanked by a pair of remarkable specialized cells called guard cells. These guard cells have the unique ability to change their shape, which precisely opens or closes the stomatal pore, acting like tiny valves.
And there can be a lot of these pores.
Huge numbers. Stomatal density can be truly enormous, and it varies greatly between species and even between the upper and lower surfaces of a single leaf. For example, the text mentions a black oak leaf can have around 405 stomata per square millimeter on its lower epidermis, underside, but absolutely none on its upper epidermis. They're usually more numerous on the cooler, more shaded underside.
How do the guard cells open and close the pore? What makes them change shape?
It's ingeniously driven by changes in turgor pressure within the guard cells themselves. Turgor pressure is the internal water pressure pushing against the cell wall. When the guard cells actively accumulate solutes and take up water by osmosis, they become turgid, swollen, and stiff. This causes them to bend outwards, opening the stomatal pore between them. When they lose solutes and water flows out, they become flaccid limp, and the pore closes.
And what solutes do they pump in and out?
The primary driver is the active transport of potassium ions, K+, into the guard cells. This pumping is powered by a proton pump, an H+ ATPase, in the guard cell membrane, which seems to be strongly activated by blue light, a component of sunlight. As K+ ions accumulate inside, they make the water potential more negative, drawing water in osmotically. Chloride ions, Cl, often accompany the K+ ions to maintain electrical balance. Malate, an organic anion produced within the guard cells, also contributes significantly to the solute potential. Interestingly, the text notes that K+ accumulation seems to dominate stomatal opening in the morning, while the accumulation of sucrose, produced either by photosynthesis within the guard cells or imported from elsewhere, becomes more important for maintaining stomatal opening in the afternoon.
And the structure of the guard cells is important too. Something about microfibrils.
Absolutely key. The guard cells have a unique structural feature. The cellulose microfibrils within their cell walls are predominantly arranged radially, like the steel belts in a radial tire, oriented circumferentially around the pore.
How does that help?
Think of two elongated balloons glued together at their ends, with strips of strong, inelastic tape wrapped radially around each balloon. As you inflate the balloons, analogous to guard cells becoming turgid, the radial tapes prevent them from expanding much in width. They are forced to lengthen, and because they are attached at the ends, they have to bow outwards, away from each other, creating that slit-like pore opening between them. Without that specific radial stiffening, they'd just swell up like sausages and wouldn't open the pore effectively.
Neat. So stomata usually open during the day for CO2 and closed at night.
That's the general pattern for most C3 plants, yes, primarily to conserve water when photosynthesis isn't happening. However, the text points out an interesting exception. Many trees and shrubs actually exhibit significant nocturnal transpiration, losing a noticeable fraction of their total daily water during the night.
Why would they do that? Isn't that wasteful?
It seems counterintuitive, but there might be benefits. One hypothesis is that maintaining some water flow at night, when the evaporative demand from the air is much lower, might enhance the transport and uptake of nutrients, like calcium, which move primarily in the xylem stream. It's an area of ongoing research.
Okay, so water enters the roots, moves up the plant, and most of it evaporates out the leaves through stomata. But how does it get up the plant? Especially in a really tall tree. That seems to defy gravity.
It really does, and this question puzzled botanists for generations. We can easily trace the pathway. If you put a cut stem or even a celery stalk in colored water, you'll see the dye drawn up specifically within the xylem tissue, clearly delineating the water transport pipes. But what provides the force?
It's not pumped from the bottom?
Not primarily, no. The leading scientific explanation, and the one strongly supported by evidence, is the cohesion tension theory.
Cohesion tension. Breakdown, please.
Okay, it starts with transpiration from the leaves, that evaporation we just discussed. As water evaporates from the moist surfaces of cells inside the leaf, specifically the mesophyll cells surrounding the air spaces, The surface film of water retreats into tiny pores in the cell walls, creating surface tension. This surface tension creates a negative pressure, or tension, essentially a pull on the water within those cell walls. This tension pulls more water from inside the cells to replace what evaporated. This, in turn, lowers the water potential within those cells, causing them to draw water from adjacent, more saturated cells, and ultimately from the water-conducting xylem vessels and tracheids that terminate nearby in the leaf veins.
So the evaporation creates a pull at the top.
Exactly. And here's the cohesion part. This tension, or pull, generated by evaporation in the leaves, is transmitted all the way down the continuous column of water within the xylem, potentially for hundreds of feet in a tall tree, all the way to the roots.
How is it transmitted so far?
Because of the extraordinary cohesiveness of water molecules. Water molecules stick strongly to each other due to hydrogen bonding, forming an unbroken, chain-like column within the narrow xylem conduits.
Like a rope made of water.
That's a good way to think of it. The tension at the top pulls the entire rope of water upwards. Water molecules also stick to the polar walls of the xylem cells, adhesion, which helps counteract the force of gravity. So, in essence, transpiration itself, driven by the sun's energy evaporating water from the leaf, generates the physical force, the tension that pulls the continuous water column up from the roots through the xylem. The plant doesn't actively pump the water using metabolic energy. It passively lets the physics of evaporation and water cohesion do the heavy lifting.
Is there direct evidence for this tension? That the water is being pulled, not pushed?
Yes, there's compelling evidence. Scientists can use a clever device called a pressure chamber, sometimes nicknamed a pressure bomb, to directly measure this tension. They take a cut twig or leaf, seal it into the chamber with the cut end protruding, and then slowly increase the air pressure inside the chamber until water is just forced back out of the cut xylem surface. The amount of pressure needed to do this is equal in magnitude, but opposite in sign, to the negative tension that existed in the intact xylem before it was cut. Measurements consistently show significant negative pressures, tensions, in the xylem of transpiring plants, especially during the day.
So the water inside is literally stretched, under tension. Doesn't that cause problems, like air bubbles?
It does. That's a major risk of this system. Water under tension is in a metanostable state, meaning it's vulnerable to breaking, forming an air bubble, a process called cavitation or embolism. If an embolism occurs, it breaks the continuous water column in that particular xylem vessel or tracheid, rendering it non-functional for water transport. Think of it like getting an airlock in a pipe.
How do plants deal with that risk?
They have several adaptations. First, the roots act as filters, preventing particles that could nucleate bubbles from entering the xylem. Second, the very narrow diameter of xylem vessels and tracheids helps stabilize the water column against cavitation. Third, if an air bubble does form in one conduit, the structure of the connections between conduits, the pits, helps prevent the air from easily spreading to adjacent functional conduits. The surface tension of water spanning the tiny pores in the pit membranes usually contains the air bubble within the embolized vessel.
Like a surface tension valve?
Essentially, yes. And in conifers, like pines and firs, the pits have an even more specialized structure called the torus margo pit. It has a thickened central region, the torus, surrounded by a porous membrane, the margo. If a pressure difference occurs due to an embolism on one side, the flexible margo allows the torus to shift and effectively seal the pit aperture, like a tiny check valve, preventing the air bubble from spreading.
Clever plumbing.
Yeah.
Any other evidence for the tension?
Another fascinating piece comes from very precise measurements of tree trunk diameter. Scientists have found that trunks actually shrink slightly in diameter during the day, when transpiration rates are highest and xylem tension is greatest. Then at night, as transpiration slows and tension relaxes, the trunks swell back to their original diameter. This subtle daily expansion and contraction directly reflects the changing negative pressures within the xylem's water columns.
Okay, so cohesion tension explains the upward pull.
Yeah.
Where does the water actually get into the plant? Root absorption.
Yes, the primary function of the extensive root system is, of course, to absorb the enormous quantities of water required to sustain that transpiration stream. Most of this water uptake occurs through the younger parts of the root system, especially through the vast surface area provided by root hairs.
Root hairs again. You mentioned that rye plant.
Exactly.
Yeah.
Those tiny epidermal extensions dramatically increase the absorptive surface area. The text mentions a 4-month-old rye plant might have over 10,000 kilometers of total root length and literally billions of root hairs, creating an astonishingly effective network for soaking up water from the soil.
How does water move from the soil through the root to the xylem in the center?
Once absorbed by a root hair or an epidermal cell, Water moves across the root's cortex towards the central vascular cylinder where the xylem is located.
Hmm.
It can travel via three potential pathways. The apoplast pathway, moving through the interconnected network of cell walls and intercellular airspaces without crossing any cell membranes. The symplast pathway, moving from cell cytoplasm to cell cytoplasm through tiny connections called plasmodesmata, effectively staying within the living continuum of the plant. Or the transcellular pathway, moving across cell membranes, potentially passing through the vacuole of each cell along the way. In reality, water likely uses a combination of these pathways through the cortex. However, there's a critical checkpoint.
The Casparian strip.
Precisely. The innermost layer of the cortex is the endodermis. The radial and transverse walls of endodermal cells contain a band of waterproof, waxy material called the Casparian strip. This strip effectively blocks the apoplastic pathway, acting like mortar between bricks. It forces any water and dissolved minerals traveling in the apoplast to enter the cytoplasm of an endodermal cell, cross a membrane, before they can reach the vascular tissue.
Why force it into the cell?
This ensures that the plant has selective control over which dissolved mineral ions actually enter the xylem stream. The cell membrane acts as a selective barrier. It prevents unwanted or toxic substances from simply flowing passively into the plant's plumbing system along with the water. It's a crucial regulatory step.
Okay, can roots ever push water upwards? Root pressure.
Yes. Under certain conditions, roots can generate positive pressure in the xylem. This typically happens when transpiration rates are very low or absent, like at night, especially if the soil is moist and warm. Under these conditions, root cells continue to actively pump mineral ions into the xylem in the vascular cylinder. This accumulation of solutes makes the water potential inside the xylem much more negative than the surrounding soil water. Water then moves into the xylem by osmosis, following this water potential gradient. Since water is essentially incompressible, this influx builds up positive hydrostatic pressure within the xylem root pressure that can force water upwards.
Is that the same as guttation, seeing water droplets on leaf tips?
Yes, guttation is the visible manifestation of root pressure. When root pressure is high enough, it can force liquid water out of the leaves through specialized pores called hydothodes, which are usually located along the leaf margins or tips. You often see these droplets on grass blades or leaves like ladies' mantle on cool, humid mornings when transpiration is low.
But this isn't the main way water gets up tall trees.
Definitely not. Root pressure is typically quite weak, only capable of pushing water up maybe a few meters at most. It's certainly not strong enough to push water to the top of a tall tree. Furthermore, root pressure is absent entirely in many plants, like conifers, and it often disappears even in plants that can generate it when transpiration rates become significant. It's more of a consequence of active ion transport into the xylem under low transpiration conditions, rather than the primary driver of long-distance water transport.
Got it. What about hydraulic redistribution, roots moving water around sideways?
Yes, that's another fascinating capability of root systems. Hydraulic redistribution, sometimes called hydraulic lift, is the passive movement of water from wetter soil regions to drier soil regions through the plant's root system. For instance, at night, when stomata are closed and transpiration stops, deep-rooted plants growing in soil that's moist deep down but dry near the surface can absorb water with their deep roots, transport it upwards through the xylem, and then release it from their shallower roots into the drier surface soil.
So they water their own upper roots, or even neighbors.
Both. It can benefit the plant itself by keeping its shallower roots hydrated, allowing them to survive and remain active for nutrient uptake when the surface soil dries out. It can also benefit less deeply rooted neighboring plants by providing them with a source of water they couldn't otherwise reach. It can also work in reverse, moving water downwards if the surface is wet and deeper layers are dry, or horizontally. It's a significant process that can reduce water stress, prevent waterlogging, and in large-scale ecosystems like the Amazon rainforest, it's thought that this hydraulic redistribution by deep-rooted trees can significantly impact regional hydrology and climate by moving large amounts of deep soil water back towards the surface where it can evaporate or be used by other plants.
Plants as plumbers for the whole ecosystem. Okay, let's quickly touch on the nutrients again. How do they get absorbed with the water? Is it passive?
Mostly not passive. The uptake of inorganic mineral ions by roots is generally an active, energy-dependent process. This is clear because if you deprive roots of oxygen, needed for respiration to make ATP, or if you keep the plant in the dark, so the shoot can't supply sugars to the roots for energy, mineral uptake rates drastically decrease, much more than water uptake does.
So they use energy to pump ions in. Where does this happen?
Most ions seem to travel predominantly via the symplastic pathway, once inside the root. meaning they cross a membrane to enter the cytoplasm of an epidermal cell or root hair and then move cell to cell through plasmodesmata towards the center. This requires at least two key active energy-dependent transport steps. First, the initial uptake across the plasma membrane of the epidermal cells, getting into the simplest, and second, the active secretion or loading of ions from the surrounding parenchyma cells into the xylem vessels themselves, crossing another membrane to exit the simplest. It's a carefully controlled two-step active transport process for most ions.
And mycorrhizal fungi help here too.
Immensely. As we discussed, those fungal partners extend far into the soil and are particularly good at absorbing and delivering nutrients that are relatively immobile in the soil, like phosphorus, zinc, manganese, and copper, directly to the root. The active nature of uptake is clear when you look at concentrations. Root cells can accumulate certain ions, like potassium, K+, to concentrations that are 75 to even 10,000 times greater than the concentration in the external soil solution. That definitely requires active pumping against a steep gradient.
Once the ions are loaded into the xylem, they just travel up with the water.
Primarily, yes. They carried upward in the bulk flow of the transpiration stream. But, and this is important, it's not strictly a one-way trip up to the leaves and out. Substantial amounts of these inorganic ions are actually exchanged between the xylem and the surrounding phloem tissue, especially in the veins of the leaves. Ions can be unloaded from the xylem, enter the phloem, and then be exported out of the leaf along with the sugars in the phloem's similate stream.
So nutrients get recycled within the plant?
Exactly. This is particularly true for the phloem-mobile ions we talked about earlier, like potassium, chloride, phosphate, magnesium, nitrogen. They are readily re-exported from older leaves via the phloem to supply younger, growing tissues. In contrast, the phloem-immobile ions like calcium, boron, and iron, once delivered to a leaf via the xylem, tend to get stuck there and are not easily re-exported via the phloem. This constant recycling and redistribution via both xylem and phloem allows the plant to efficiently allocate its limited nutrient resources to where they are most needed for growth and development.
Okay, that covers water and minerals moving up. What about the sugars moving out of the leaves? That's phloem transport or translocation.
Correct. While water and minerals ascend primarily in the xylem, the sugars manufactured during photosynthesis, mostly sucrose in many plants, move out of the leaves and are transported throughout the plant in the assimilate stream within the phloem. This process is specifically called translocation. These sugars are transported to wherever they are needed to sites of active growth, like shoot tips, root tips, developing leaves, or to storage organs like fruits, seeds, tubers, or storage roots.
And this follows a source-to-sink pattern.
Yes, that's the fundamental principle. A source is any part of the plant that is producing more sugars than it needs for its own respiration and growth, primarily mature, photosynthesizing leaves. A sink is any part of the plant that cannot produce enough sugars to meet its own needs and therefore must import them. This includes roots, shoot apical meristems, developing fruits, seeds, flowers, and storage tissues when they are accumulating reserves.
Can a tissue change from being a sink to a source?
Absolutely. A storage tissue, like a potato tuber or a beetroot, acts as a sink when it's growing and accumulating starch or sugars during the growing season. But the following spring, when the plant starts to regrow, that same storage organ becomes a source, breaking down its stored reserves and exporting sugars via the phloem to fuel the growth of new shoots and leaves. Source-sink relationships are dynamic and can change depending on the plant's developmental stage and environmental conditions.
And sinks compete with each other, like fruits.
They do. Developing fruits are often very strong sinks, drawing heavily on the sugars produced by nearby leaves, and sometimes even leaves quite far away on the plant. There's often competition between different sinks. For example, multiple fruits, or shoot growth versus root growth, for the available supply of sugars. Hormones often play a role in mediating this allocation.
How do scientists study this sugar movement? Radioactive tracers again.
Yes, radioactive tracers have been invaluable. By supplying a leaf with CO2 containing the radioactive isotope carbon-14, 14C, the sugars produced during photosynthesis become labeled. Scientists can then track the movement of this labeled sucrose throughout the plant using autoradiography or by sampling different tissues over time. These studies definitively confirm that translocation occurs within the sieve tubes of the phloem.
And aphids help too, the insects.
Aphids turned out to be surprisingly useful, almost like natural microsyringes. These small sap-sucking insects have incredibly fine needle-like mouthparts called stylets, which they can skillfully insert directly into a single phloem sieve tube to feed on the sugar-rich sap.
They tap right into the pipe.
Exactly. And because the sap inside the sieve tube is under positive pressure, we'll get to why in a moment, it actually forces the sap out through the aphid stylet. Sometimes it comes out the other end of the aphid as a sugary droplet called honeydew. Scientists learned they could carefully cut the aphid away, leaving just the stylet embedded in the sieve tube. And pure phloem sap would continue to exude from the cut end for hours, driven by the plant's internal pressure. This allowed them to collect and analyze the composition of phloem sap directly.
What did they find in the sap? Mostly sugar.
Overwhelmingly sugar, yes. Phloem sap is typically 10–25% dry matter by weight, and over 90% of that dry matter is usually sucrose in most species. But it also contains smaller amounts of other things: amino acids, proteins, various RNAs, plant hormones like auxin traveling from leaves, and some inorganic ions being recirculated.
And it moves fast.
Remarkably fast for diffusion through living tissue. Measurements using tracers or aphid stylets show that sugars can move longitudinally through sieve tubes at rates of 50 to 100 centimeters per hour, sometimes even faster. That's much faster than simple diffusion could account for over those distances.
So what drives this rapid long-distance movement? Is there a theory for phloem transport like cohesion tension for xylem?
Yes, there is. The most widely accepted explanation is the pressure flow hypothesis, first proposed by Ernst Munch in the 1930s. It elegantly explains phloem transport as being driven by osmotically generated differences in turgor pressure between source and sink tissues.
Pressure differences again. How does it work?
Let's break it down. It starts at the source, typically a photosynthesizing leaf cell. Sugars produced there are actively loaded into the sieve tube companion cell complex in the smallest veins of the leaf. This loading process dramatically increases the solute concentration inside the sieve tube element.
Making the water potential very negative inside the phloem at the source.
Exactly. This highly negative solute potential causes water to move rapidly by osmosis from the surrounding xylem and other nearby cells into the sieve tube element at the source. This influx of water generates a high positive turgor pressure within the phloem at the source end.
Okay, high pressure at the source. What happens at the sink?
At the sink, say, a growing root cell or a developing fruit, the opposite happens. Sugars are actively unloaded from the sieve tube element and consumed or stored by the sink cells. This removal of sugar decreases the solute concentration inside the sieve tube at the sink end.
Making the water potential less negative there.
Right. As the solute concentration drops, the water potential inside the sieve tube at the sink becomes less negative, higher than the surrounding cells. So water moves by osmosis out of the sieve tube at the sink, flowing back into the xylem or adjacent cells. This outflow of water results in a lower turgor pressure within the phloem at the sink end.
So you have high pressure at the source and low pressure at the sink.
Precisely. This difference in turgor pressure between the source and the sink creates a pressure gradient along the sieve tube. And just like water flowing through a pipe from high pressure to low pressure, this pressure gradient drives the bulk flow of the entire phloem sap solution water and dissolves sugars together from the source towards the sink.
The sugar molecules are just carried along passively in the stream?
Essentially, yes. They are carried along by the mass flow of water driven by the pressure gradient that was generated by the loading and unloading of the sugars themselves. The water that leaves the phloem at the sink then largely recirculates back to the source via the xylem, completing the cycle. It's a beautiful, self-contained system driven indirectly by the energy used for active loading and unloading of sugars.
Okay, but how do the sugars get into the phloem at the source? Phloem loading, are there different ways?
Yes. The mechanisms of phloem loading, getting sugars from the photosynthetic cells into the sieve tube companion cell complex, can vary among plant species. There are two main routes described. Apoplastic loading is thought to be common in many herbaceous plants and crop species. In this mechanism, sucrose produced in the mesophyll cells first moves through the cell walls and intercellular spaces, the apoplast, to the vicinity of the phloem. Then it is actively transported across the cell membrane into the sieve tube companion cell complex. This active transport step requires metabolic energy, ATP, and usually involves specific transporter proteins, like an H+ ATPase, proton pump, creating a proton gradient, and then a sucrose proton symporter protein that uses the gradient to move sucrose into the phloem against its concentration gradient.
So it pumps sucrose in, concentrating it. What's the other way?
The other main route is symplastic loading. In this case, sucrose moves entirely within the living continuum, the simple S, passing directly from the mesophyll cells through plasmodesmata into intermediary cells, a specialized type of companion cell, and then into the sieve tube elements, without ever entering the apoplast.
How does it move against a gradient then? Or does it?
Well, in some plants that use symplastic loading, especially in some families like the cucurbitaceae, squash, melons, there's a clever biochemical trick called the polymer trapping mechanism. In the intermediary cells, the sucrose that arrives from the mesophyll is quickly converted into larger sugars, like raffinose, a trisaccharide, and stachylose, a tetrasaccharide, by adding galactose units. These larger sugar polymers are too big to easily diffuse back through the narrow plasmodesmata, leading back to the mesophyll. However, the plasmodesmata leading forward into the sieve tube element are wider, allowing these larger sugars to pass through.
So they trap the sugar in a bigger form.
Exactly. It creates a one-way flow, effectively concentrating sugars in the phloem by continuously converting the incoming sucrose into larger molecules that can't leak back out the way they came in. Our source illustrates these intermediary cells in the minor veins of Fuchsia triphylla, showing their characteristic, numerous plasmodesmata connections needed for this pathway.
Are there other variations?
Yes, the text also mentions that many tree species seem to use passive symplastic loading. In these cases, sugar concentrations might actually be fairly similar between the mesophyll cells and the sieve tubes, suggesting that loading might occur more passively down a concentration gradient without an active concentrating step. In some species, like the basal flowering plant Amberella, or common plants like Coleus and Cucurbita, which uses polymer trapping but maybe also other methods, might even be capable of using both apoplastic and symplastic loading pathways, depending on conditions. It shows there's remarkable diversity and flexibility, even in this fundamental process of loading the plant's food transport system. Hashtag outro. Wow, what an incredible journey that was, really diving deep into the hidden physiology of seed plants. We've seen how this complex array of chemical hormones meticulously orchestrates everything from the microscopic dance of cell division and elongation right up to the grand processes of fruit ripening and root development. We explored the truly ingenious ways plants sense and respond to their environment: light, gravity, touch, day length, temperature, fine-tuning their growth, and precisely timing their vital life stages. It's almost like they possess senses, just not in the way we typically think of them.
Yeah, and we didn't stop there. We really dug deep into how plants acquire those essential nutrients from the often challenging soil environment, decoding the complex ways these elements cycle through ecosystems, and discovering the ingenious strategies they employ, like partnerships with fungi or specialized roots to overcome deficiencies and even cope with toxicities. Finally, we followed those incredible internal transport highways, the xylem and phloem. We saw how xylem moves vast quantities of water seemingly against gravity thanks to the elegant physics of the cohesion tension theory, and how phloem transports sugars via the brilliant pressure flow mechanism, all while showcasing the plant's remarkable adaptability to the constant challenges of life on land. From the minuscule amounts of hormones dictating overall shape to the vast flows of water sustaining entire forests, plants are truly intricate masters of biological design.
This whole deep dive really makes you pause and think, doesn't it? If plants possess such sophisticated, often completely silent, systems for sensing their world, communicating internally, and adapting dynamically, what other hidden biological languages are still out there in plants or other organisms that we are yet to fully decipher? And maybe more profoundly, what lessons might these quiet masters of resilience and adaptation still hold for us, if only we learn to observe and listen more closely to the non-human world around us?
That's a great thought to end on. Thank you so much for joining us on this deep dive. We hope you've enjoyed this fascinating exploration of the physiology of seed plants, based purely on Section 6 of Raven Biology. Maybe you'll look at the next plant you see, whether it's a house plant or a giant tree, with a little bit more awe and understanding. This has been a deep dive from the Last Minute Lecture team.